a complex society. So certainly global warming may create the threat to our population of certain viruses, but there's travel involved, poverty, war, multiple things that also add to that. But I think global warming is the topic of the day, and certainly what may be happening that it may initiate the initial threat. But those other factors are certainly going to be involved. So where will the next pandemic arise? We all know about Ebola virus. It's not really a pandemic, and there's a reason, and there's a point I put this in. Ebola virus kills very quickly. The incubation period is 2 to 21 days, something like that. And the patient is not infectious until really they're symptomatic. And then until they're really, really symptomatic, and they're grossly infectious. And the majority of people that physically come in contact then with these patients will get the disease, and a certain large percentage will die, although we've made progress recently in Ebola virus. But the point is, it's probably not going to be the next major threat because most of the patients die. It will be a threat locally, and maybe a large number of patients will die in that local situation, such as the recent outbreak in West Africa, which I think was the worst. But it's hard to become a global pandemic because it's pretty easy to screen anybody who's remotely sick when you know there's an outbreak, doesn't come into the United States, Western Europe, etc. So it's a large epidemic. And it came from the bats, and as far as we know, fruit bats. And fruit bats are sort of the natural Ebola host virus, and it also was introduced to humans through close contact with any kind of body fluid. Because of this long incubation period, like I said, it's just probably not going to be a major threat because humans aren't infectious until they're symptomatic. Whereas if you compare that to the HIV, the patients are highly infectious, no symptoms, don't know they have the disease back when we first found the disease and diagnosed it, and to date. And another pandemic that we already have, we all know about, the Zika virus, and it's here, and it's really not, it's actually becoming pandemic. And the interesting thing is that the mosquito that causes this disease bites during the day and night, unlike the mosquitoes that cause malaria, so an additional problem in controlling the disease. We all know about the multiple birth defects, and we have local Zika transmission in the continental United States. So it's entering the pandemic phase. It may attack a certain patient population and not be a massive pandemic, but it's already becoming a pandemic. So where is the next threat going to arise, the canary in the mine? Probably in the experts in the field are saying probably in South America, perhaps Southeast Asia, parts of Africa. These are all very biodiverse areas. They are home of thousands of species, and they are warming, and that increases perhaps the species. Also, other factor is people coming into zones that they didn't inhabit before and coming in contact with various species. And so it's the tropical countries that are most likely to harbor the next threats. In all of these countries, it's a hotbed of viruses. If you think of viruses, that every mammal probably has 20 or 30 viruses times the number of mammals, which is huge. So you have hundreds of thousands of viruses. And things like bats are probably the biggest threat of carrying viruses. In the United States, it's rodents, primates in other countries. The source of the Zika virus. So there's a huge potential of one of these viruses becoming pandemic. And I'm just going to say that we know about all the other viruses. I'm talking about the viruses we don't know about. So I was going to put on a slide the viruses at risk that might be important for us to watch. And actually on the list is 600 that we know about that may be threatening. And so entirely different concept right now. This is north, and this is melting. We all know that, part of global warming. As the glaciers melt and the tundra melts, and just think of Russia as being hundreds and hundreds of thousands of square miles of tundra, we're exposing ground that hasn't been exposed in thousands of years. And these viruses and bacteria can live in these conditions, dark, cold, wet, and survive for thousands and thousands of years. And just recently in Africa, in Russia, there were a large variety of cases of anthrax, from anthrax being recovered once the ice melted. So we don't know even what organisms are here. We haven't been exposed to them for thousands of years, so we don't know what new disease we may see. And I think the key to what I'm trying to say is that we know about TB, we know about these others. We've done a phenomenal job of at least controlling HIV. It's going to be kind of hard to identify and control what we don't know yet. And so, I think I mentioned anything there. So I want to just conclude my talk and say that all of us here probably enjoy a very good healthcare system. And a lot of the world doesn't. And we've just heard from our speakers here whose life's work is trying to make the rest of the world as safe as it is here. So I'm just going to, I presented this slide because this may be an advanced medical center in certain countries. We think of Mass General, New York Presbyterian, all of the medicines that we have. In 1969, courtesy of the U.S. government, I had a year tour in Vietnam. And so I did a lot of tropical medicine. I was a battalion surgeon, saw tons of trauma, but also a lot of infectious disease. And we literally were in a rainforest jungle, sometimes raining, and we would have people line up 200 or 300, after our troops went in and did what they did, we would have a medical civil action program, and we would treat 200 or 300 people in the village who'd never seen a doctor. So, I mean, these weren't huge villages, almost everybody in the village. And we saw phenomenal diseases. I felt bad we could only do so much, but we had 4 medics, myself, and I just remember vividly a little kid had an infection on his head that probably had a gallon of pus on his head. He was near death. And all we did was lance it and gave him a shot of penicillin. We were back in 2 weeks and he was alive and well.
Okay, so
didn't have a resistant organism, but they don't. But and when he saw us, he had a bunch of banana leaves or something wrapped on his head. So there are lots and lots of areas of the world that even today, unfortunately, medical care that we talk about here and take for granted is not that great.
Okay, Dr. Lutz, we have to move on because Jules has another meeting afterwards.
And so the next slide, I want to leave you with this: conflicts, areas, and infectious disease. Standard bugs that are not a problem will become a problem in conflict areas. So salmonella, simple bugs that we don't get, kill a lot of people. And my final statement from Vietnam is our favorite saying. Thank you.
Okay, thank you very much. Okay, our next speaker is Dr. Jules Mitchell. He's the president and co-founder of Target Health, a New York City-based full-service ECRO dedicated to all aspects of drug and device development, and he's going to be speaking about the repurposing of existing drugs.
Okay, so you got it going? Okay. Mitchell. Sorry. Mitchell. I point to the screen, but he can't see that, you know. So. That looks good. You got the clicker? Yeah. Okay. So hello, everybody. Just it's interesting because before I got into the pharmaceutical industry, I had an eclectic job, but one of the things I was doing was mosquito control for malaria. So I was the fish guy because the fish ate the mosquito larvae, and that was one of the better ways to do it. So it's nice to come around. So today we're going to talk about repurposing drugs, and based on the wonderful presentation from WHO and the World Bank, and it was nice to hear that they're qualifying manufacturing sites so we can really efficiently do things. So we'll talk about repurposing drugs, and I can tell you that even in the developed world, even this morning and yesterday we had 2 conversations, one for repurposing drug this morning for Tourette syndrome, existing drug, and yesterday we're on the phone, that's why I have to leave a little early today, with a surgeon who's actually developed some old drugs for PTSD, and he claims he does one injection and the PTSD disappears, and these are all old drugs, and we'll talk about some of them today. So this is a hot area. The problem is, and I think the World Bank and WHO has to deal with it, is the pricing, and we won't get into that today, but how to make these things available to people when there's no necessary and economic motive to develop them or even to sell them. And I think that's where one of the areas where government, I think, could do a wonderful make a wonderful contribution. I hope I hit the right button. Good. So we were asked to do a couple of questions. So we'll talk about what are the advantages of developing a drug for a new indication. Again, we'll talk about the regulatory space and the clinical trial space. What are some of the regulatory hurdles? And we'll talk more about the UN. And again, I want to comment about that WHO can, with their, obviously their leverage, they could actually bypass some of the regulatory hurdles, especially within countries. And I think will make things, very attractive. And then what's the best way to get regulatory approval? And we'll talk about some of these things. So we're going to focus. It's a little different than the global stuff, but I think if we don't get the drugs to the market and we don't understand how they work, we really shouldn't use them. So just very briefly, so we're a full-service CRO, which means we're a contract research organization. We help companies, mostly small companies, take products to the market. And what's also interesting is we also built some software that allows a paperless clinical trial. So this may be some advantages to the developing world, as long as you can get internet. Okay. Whoa. Okay. Oh, it's one of my dynamic slides. So the other thing is that we've gotten products to the market for pancreatic enzymes, for CF, we did prostate cancer, head lice, which is an interesting. There's another product that was developed. This was and it's an approved product now. That was benzyl alcohol, a very simple chemical that's been around forever. And there's a product that within 1 day kills all the head lice. So that's also a problem, in the developing world. Okay? And then we've also worked periodontal disease, hair loss, et cetera. So, and the idea is how to get products to the market. So let's talk about repurposing drugs. So what does it really mean? So it really means taking a product that exists for some other reason. It's been approved somehow, somewhere, and then we're gonna switch it to some new indication, new formulation, new approach. We could take an oral drug and do it topically. We could do an IV drug and make it sublingual. We could do all kinds of things with it. And just some examples. So now you have aspirin. Everybody knows about aspirin now. Everybody's using it take it at night or in the morning to prevent clots. We talked about AZT. Was developed originally for cancer. And what's interesting also is that there are several products out now. One in Alzheimer's that's being developed. That was originally developed by NIH, I think, for cancer, and they found another mechanism of action. So the good part about pharmaceuticals is that you're dealing with a chemical company, and we were talking about diagnostics earlier. And I think one of the futures of the pharmaceutical industry, it will be diagnostics, because we're going to find out the mechanism of how things work, or some disease works, and then we'll find the drug that's already approved and treat. Like this is going on now with SIV. There was a simian virus equivalent to HIV. The NIH did some studies and found that these monkeys responded to a certain monoclonal antibody. They figured there's a similar monoclonal antibody that's out there for a GI indication, and they're doing studies now in humans to see if it actually not just puts the virus in quiescent stage, but also actually kills the virus. Okay? Viagra was developed originally as an antihypertensive, because it's an alpha blocker. Thalidomide, which everybody knows caused the terrible abnormalities in the newborn, is now for multiple myeloma and epileptic drugs. Okay. So one of the advantages of why we do it, number one, is you got to do toxicology and see how things work, so we can eliminate a lot. We can't probably eliminate all the toxicology, but a lot of it. And we'll give examples later. So the drug substance is the same, so we don't have to worry about the chemical itself. Okay. Also, that from the business point of view, a new indication or sorry about the typo there, new formulation may provide some IP protection. So the people here in the business world, they all want to make some money. So unless the government takes over, IP is important. And I think government actually, in this case, can play a very important role. It's much less cost to get to the market and can be more affordable in the marketplace. So I think that even if companies don't want to do it, if the government, I think actually one model is for the government or governments to actually create manufacturing plants. There are a lot of them out there now, and actually make some of these drugs and support them and make them available at reduced costs, because a lot of people aren't. One of the reasons we have drug shortages now is that the prices are too low. This is supply and demand stuff, okay? So some of the FDA issues. So the FDA allows, it's something called a 505(b)(2), that's in the Food Law, Food, Drug, and Cosmetic Act, and that's a section. 505(b)(1) is a new chemical entity. So what it means is that you can use data. So a B2 application is one where some of the investigations, some of the studies relied on the applicant's approval, were not conducted by the applicant themselves. So normally when you develop a drug, you do all the work yourself. But there may be other people have done some work on it, and we're doing things now based on the literature, et cetera. So examples could be published literature and even FDA findings of safeness and effectiveness. So, that's why whenever academia does a study, we try to make sure it's somewhat robust, because we can often use that data to help get FDA at least comfortable that the drug is safe. So when we work with FDA, when you're doing these concept of repurposed drugs, or somebody has an idea even to develop a new drug, or they have a new indication, and they think this is an old drug, so we always tell people, meet with FDA early in the process. Always bring the experts, like we have in the room here. Okay, bring some experts with you. We also tell them now, even to bring, though it may not be feasible for some of these studies, but some of these indications, but even bring patients now, because the patient voice now becomes very important. What's actually important to the patient, not just to the doc and some measurement. And also, if you have a plan, you can't go say, FDA, how do you do this? You have to tell them what you want to do. And actually, if you do it right, I always tell people, FDA will give you some free consulting. You may not like everything they tell you, but at least you know what the rules will be. And I'm going to give you a couple examples. So here's a drug where multiple sclerosis is a chronic disease, okay? And we don't have to say what it is, but most of you will know what it is. So some people have been studying statins for some reason, okay? And this is all published, so we're not sharing any secrets. So so here's Lipitor. So now it's a generic drug, okay? Can be made cheaply. It's a small molecule, not a big deal. So in the new study on the bottom, it says people with early forms of MS, the statin drug Lipitor cut the risk of developing new brain lesions by about 50% compared with placebo. New lesions are reliable indications of future MS attacks, according to the study. So that means maybe even some of the other statins will work, okay? But there's an area where if you have perhaps a very low-cost drug, get into an area where, into developing country, make a deal with someone who's making this thing and give it out to the people. And you know, the other thing you could do is actually do studies in developing world if you actually do them correctly and clearly ethically. Okay? So another example, we're working on this one now. This is Dravet syndrome. So we're not showing the mosquito, but this is zebrafish larvae, okay? And actually I did work in fish for my PhD, and my mentor was a zebrafish expert. So it was nice to see it. Now they put genes in zebrafish, okay, and they put drugs. What they do is they put drugs in the water, and that's how they found things. So this was actually came out of NIH. So Dravet syndrome is an early epilepsy, starting about the age of 2. Pretty nasty business, and so at UCSF, University of California, San Francisco, they screened 320 compounds, just throwing it in the water, okay? Not too complicated. And they found that clemizole, which was a drug that was developed by Pfizer in the early 60s, okay, was effective in inhibiting the seizures, okay? They claim clemizole was approved by the FDA, but it's a very old drug, and we can't seem to get the data from them. But it said that based on what they know about clemizole, they couldn't predict it, but they just screened a whole bunch of drugs in a living organism. And for this one, we actually got orphan drug designation by FDA, and we're starting studies very soon. So there's an example of how we can take existing products, probably with relatively low cost. And I think in this case, if we're dealing with developing countries, I think with some government support, I think you could do amazing things, especially if we're finding companies that don't want to develop this thing because of the price, and we all work together, get these things approved, because it's not that complicated to get these things approved, and then just find somebody to make these drugs. Maybe some of these plants that have just been approved by, cleared by WHO, would be more than happy to do it, you know, and because a lot of these small molecules are actually very inexpensive to make. So, and if no, and the good part is, because I am a capitalist, if we actually can find, if we find that nobody wants to do this, then that's a wonderful opportunity for everybody to work together to get this done. Okay, so thank you very much.
Thank you, Dr. Mitchell, for your talk. Dr. Mitchell has to leave in a minute or two. If there's one question, burning. Yeah, one burning question. And please state your name and your organization.
Please.
Hi, Dr. Mitchell. My name is Garcia, a people entity Rainbow and on the board for Tablet Pioneer City Chapter of the United Society. So thanks for your talk. My question is, what markets are you seeing the drug repurposing being where there's the biggest appetite? And how are the governments approaching you? Are they finding you or are you finding them?
Yeah, so I think the governments don't exist right now in doing that. Was a proposal to the world, you know, to do this. I think the repurposing of drugs right now is more people have intellectual property on the formulation itself, because if it's just the neat drug, anybody can use it. So they're usually, we found with some of these are the people who actually have a passion or they have the disease, like a drug we're working with in MS, not just Lipitor, but some other things, is actually someone who had MS, did his own research, he's a lawyer in Italy, did his own research and treated himself. Because he said, oh, you know, somebody did a study here, somebody, and that's not uncommon, by the way, to find this. So, but right now, I think it should be a movement. It's not yet, but I think it's doable, because one of the most difficult parts is finding the funds to develop something where somebody, where the you know, the investors aren't, there's not going to be a big return on investment. In fact, I spoke to a major, I know a head of neurology at one of the big pharma companies, and he's a really good guy, but he said, there's no market for us. If it's not a new chemical entity, we're not going to touch it. And not meant as a criticism. Yes? Burning question, that's even better.
And the opioids epidemic. No, they have a new medication, an injection for the antidote. What are your thoughts on that? They had $60 million to spend, and it has been cut in developing an antidote for the heroin epidemic, which is an act to the Attorney General right now.
So, I didn't hear the first thing you said, but did you say there's a known product?
Epidemic, as you know. Now, the injection is $2,000, and the insurance does not want to pay. It's too expensive. What are you aware, since you are a developer, and do you know anything about it? That's my question. No.
No, I don't know anything about it, and I don't know why something is $2,000, you know. It's life, you know, but I don't know what the chemical is, so I can't really comment on it.
Okay, thank you.
Yeah. And the other is Dave Luke is here, who works at our company. So, and he's formerly out of Pfizer.
Dave, can you just stand up so people can see you?
So he's also capable of jumping in and answering some questions. And if he doesn't answer it right, let me know. If he doesn't know the answer, I would like to know that. Okay. And I'm really sorry. I just had this came late, and I'm really sorry. I got to go.
No,
okay.
Thank you very much,
Thanks.
Jules. Okay, so our next speaker is Dr. Norman Gray. He is a senior biomedical scientist with 30 years of executive management experience across a wide range of healthcare disciplines. He was also named Carlton Highsmith Chair of Innovation and Entrepreneurship, and he is going to speak about the globalization of disease.
Good afternoon. We're running way behind. So I did a presentation. I did two. And the first presentation was going to be speaking about what you expect me to say. The second one, I was going to ask permission to be blunt and to tell everybody where the baby is ugly. So I'm going to go straight to that to save time, and hopefully we'll catch up. Do I have permission to speak bluntly? We can address the issue of what we should do if a major virus or bacteria, a pandemic, hits us again. We can have the United Nations. We can have a lot of the tree huggers. And I fall into that category. I'm a tree hugger. A silent one, but a tree hugger. Come up with great solutions on paper. I'm an old man. I've heard them all. I read a lot. And because I'm a tree hugger, I read the solutions that everybody's saying, and they're regurgitating them over and over. Every couple of years, put a little twist on it, put a little technology on it, but it's the same solution. I propose to you that the solution is not there, that we have to start looking at the reality. We have to look at when money is put out, whether to a developed or a developing country, that there is greed, corruption, and the money, I've watched it myself, you put $1 out, 50 cents goes to the guy who put the deal together, 25 cents of the 50 left goes to somebody on the inside who just had to get paid. And we're lucky if 10 cents gets to the project. Give the UN, give all of the agencies all the credit for trying their best to come up with solutions of how to get the dollar to the project. But it hasn't happened. I haven't seen it yet. I have seen France put a highway in Puerto Rico, put a toll on it, and that works. I've seen China brought their people from China, didn't give the money, brought the materials, brought the people, go in the country, built the roads, built the healthcare system, and that's okay. I've seen China, Taiwan, in a consortium, go to St. Lucia, build a phenomenal healthcare hospital, brand new, but they didn't donate the equipment, and the hospital remains unopened 7 years after completion. The problem is not the money, and the problem is not the ideas. I now teach a course, an entrepreneurship course, and I start the course with innovation is equal to the idea or the invention times commercialization. So we can have an idea, and if somehow we do not commercialize this idea, the commercialization goes to zero, and the innovation is zero. I propose that we stop talking. The solutions are there. They have been there. We can tweak them with technology. We can tweak them with FaceTime and, you know, all the high-tech stuff that they have today. But we have to find a way to to commercialize these ideas. Without that commercial piece, we can talk about all these pandemics and all the solutions and all the United Nations and putting it on governments. It's just going to happen the way it's always been. 10 cents, maybe, will go to the project. I watched anthrax, Bear, New Haven, Connecticut. The pandemic hit, and within 6 months, with a concentrated effort, with all the resources of the United States Treasury behind them, they found a solution. 6 months. True story. We can talk about it after. That's what we need. And therefore the challenge for me is not all these ideas. I can come up with a bunch of ideas. The challenge is, how do we make it commercially viable so that somebody will take the interest and move it forward? I'm not one of those people who believe in conspiracies, but I do believe that there are solutions to cancer, to just about everything we have. And until we stop, face reality, stop relying on governments, and look for that commercial solution. I don't have it right now. But we can look at what France did. We can look at what the Chinese are doing when they give. And maybe there's a solution there somewhere. So I'll be the bad guy in the room today.
Okay, thank you, Dr. Gray. So now we're going to move to question and answer. I'm going to just start with a few questions. The first question to Richard Lutz. So you talked about a pandemic and that a pandemic might be inevitable. How soon are experts predicting the next pandemic?
All the experts in the field,
predicting. Can we have that microphone?
So all of the experts in the field are certainly predicting a new pandemic, and there's no reason not to expect so, just from what everyone knows and what I just presented. And certainly we're seeing new pandemics now. Zika, the last year, if you're going to call it a pandemic. I would say that everyone, without giving dates, they're certainly talking about inevitable and soon. And to me, that would be the next 2 or 3 years. Global warming, as we know, is threatening us with new viruses every day, and travel is making the spread of those viruses greater. I think the key issue is what's the size of the pandemic, and not whether we're going to have it. Something that surprised me in my reading was a moderately severe global pandemic will cost the world about $500 billion. Huge sums of money. That's 0.78% of our gross domestic product. So that would be a major pandemic, and that's probably what the HIV epidemic cost if we add it all up over the years. So an epidemic, a pandemic of that size may be a little far away. Newer, smaller pandemics that we hopefully can identify and control are probably much quicker.
And what about our ability to combat pandemics? Can you talk a little bit about that?
The experience that I told you about with HIV, which is very personal, was 30 years ago. And we've just come so far in science in the last 30 years. And many of the people in this room are involved in database collection and that. I think one of the key things, first of all, is to identify the pandemic very early on. And there are certainly groups that are attempting to do that. There's a couple of groups here in New York, in fact, ECHO, I think, or something like that. And so identifying the outbreak is much more likely today than before. And our scientific progress, I read the science every day, I'm involved in it, we develop biomarkers. It's phenomenal. It's hard to even keep up with the progress. So the tools that we need are increasing so rapidly to define vaccines, treatments, improve antibiotics, etc. Unfortunately, we sort of need a stimulus of the pandemic to stimulate more research. It's sort of what Norman just said. That, you know, you've got to get the products to market, and you've got to get them commercialized, meaning available, basically, before they're going to be effective.
And that actually leads to my next question to Dr. Gray. I mean, you talked about commercialization, and this, I mean, we know that there are plenty of patents being produced, there are lots of patents, but the incentive to commercialize and getting people to commercialize, what are some strategies that you've found that have been successful that lead to commercialization?
It's a difficult one. But I would say the day that we can tie Cialis to a rare disease, everybody knows what Cialis is, right? The male, what do you call it? Enhancement, okay? The day we can tie that to microvillus inclusion disease that kills a baby on day 4 of birth, that's when we'll have a solution. Right now, I don't see it. We've got Genzyme, who is one of the pioneers in rare disease work, and Genzyme has been able to make a living, a really good living from it. But for example, if you have Gaucher and you use the Genzyme drug, it costs you $1.2 million per year. So go figure. Right? You know, how does somebody who is a Sephardic Jew that does not have the support that is based in southern Spain, how does that person afford $1.2 million?
Okay, I realize we're a little behind schedule. I'm just going to, if there are 1 or 2 burning questions. Okay, I see we have one here. Go ahead and please state your name and affiliation, and then we have another one right here.
Good afternoon. This question is for Dr. Norman Gray. My name is Jose Garcia with Angel Hack and also on the Tau Beta Pi board. So Dr. Gray, you had mentioned the importance of commercialization. It's a follow-up to Dr. Halpern's question. Do you feel that since a lot of innovations that are successful in commercialization are done by entrepreneurs as opposed to institutions, do people leaving institutions to do it on their own through accelerators like Techstars, do you feel like that's a much more viable strategy, or can we do it in an institutional context?
First, I disagree with you on the premise that an entrepreneur has to be an individual. Institutions are entrepreneurial, and I can name a lot. It's a buzzword today. When I was growing up, I couldn't spell the word. I didn't even know it existed. And, you know, my partners and I, 3 of us started a company in 1984, and we built it to $1 billion. We had no clue that we were entrepreneurs. The question really is, what is the incentive? And is the incentive to the individual or to the company is the same. What is commercialization? It means that everybody has the incentive to make a lot of money. And I don't care what the system is. You know, we call systems communism, we call systems socialism, we call systems democratic, or it doesn't really matter. Money is at the end of it. And if you can find some way of making a venture feasible so somebody can make some serious money off of it, it goes. And it goes back to the bear and anthrax. 6 months, starting from zero. They had a solution. But what did they have? A huge pot of gold at the end. And they had the resources of the Treasury of the United States.
And some of you might want to look at the I-Core model, which is through NSF. We had a question right here.
It was here.
He had asked first, and then you're afterwards.
Just skip it and go on if that's the case. But the thing that I'm curious about is what we're looking at here is global warming, and we know that that's happening. But this, I think, probably isn't the topic of this conference. But the question is why it's happening. And that one, as far as I know, at least as of a couple of years ago, even though we've talked about it at the UN and everything like that, has not been solved. Because we say, okay, it's carbon loading of the atmosphere and stuff like that, but it's so complicated a many-body problem to go from the projection to the answer on that we may never be able to solve that. We haven't done it yet. We haven't worked on many body problems that are this big yet. I wonder if any of you know where there are the workers going on this now. I mean, I've talked with people at Texas Tech and places like that, so I'm a little bit up on it, but I'm not an expert in the area.
Yeah, I mean, I think this is... Okay, this is a little beyond. I mean, the global warming and disease is really the main related. But maybe we can have a very brief answer. Also, in the future, please try to make your questions brief. Yes.
Yeah, thank you. My name is Dr. Moody. As a physician and somebody working in global health, and I'd like to congratulate the panel, especially you, Dr. Lutz. My question is, with the current administration, we are all freaking out because of the cuts. So we are talking about the P word, pandemic. So what are your suggestions is how do we work together as a global community? Because this is something we're really scared about. I mean, every day my students and my residents ask me this question, and I really don't have an answer to it. So maybe you could tell us, how do you think with your knowledge, because you kind of digressed into infectious diseases with all the work you did. So what suggestions do you have for us to see how can we address this issue? Thank you. I kept my question short.
Thank you.
A panel like this is a simple grassroots start. A small number of people, but influential people who can go back and have the message grow. I have to say that when I reviewed this topic in some detail, I became increasingly worried over what I already knew, without a doubt. And answers to what you're asking are exceedingly difficult. I do think it has to be a UN, WHO backed program, because these are global problems. These are not U.S. problems. They're not France problems. And we've seen the countries where the diseases are going to develop. So I certainly don't want to simplify it. I think that my opinion is we are, personal opinion, we're going to have another major conflict, another major pandemic, and then everybody's going to respond and say, oh my God, just like with the HIV. So everybody came out of the woodwork. There was a phenomenal amount of research. We learned millions of facts and things during the HIV epidemic. It was a learning experience. Some of my friends were involved, Bob Gallo and others I mentioned, but it's going to take the disease to mount that attack. Fortunately now we have so much basic science, it won't be that, we just have to mount the basic science that's there. So the good part of the news is that our basic science community throughout the world, throughout New York, throughout the institutions, is really doing phenomenal work on understanding disease. And now that we understand the disease, infectious disease, and cancer, we can treat it a lot better. We do need to know what we're treating, though.
Okay, now with that, I would like to thank our speakers for your excellent presentations, and also thank you for your questions. I will now give the floor to Aaron Etra.
Yes, thanks very much to our panel and to our moderator. We're going to go right into the next panel. Those of you who want something to drink, you've found, I think, that there is some in the back there, so help yourself. But we want to start and keep along in our program. So with the next panel and our wonderful moderator, Dave Luke, please come forward, and I will give the floor to him. to introduce our next panel of speakers and ask the panelists, of course, to take their place at the table. Yeah, you get your name.
Do you want us to sit up here? Yeah, please. Then I gotta watch if I don't pick my nose. Well, just don't spit on it.
While our panel is getting together, the one takeaway I think that we all need to have in this discussion. So please do try to take your drink and come and sit down as quickly as possible. Of course not. Okay.
I'll call him back.
Okay, great.
I'm Rick Flat. Hey, Barry, how you doing? Hey, Steve, how you doing? Good. That's David. Hello.
You barely stand up.
You can introduce us?
Do one line.
It's just so they know your name and they at least know they have an idea. That's you? Are you going to be on the...
Right here. But I'm first up, so I might...
You're the moderator.
No, I'm speaker. I'm the moderator.
Okay, all right. Usually the moderator... Well, so wherever you like it. That one's last.
Yeah, that's cool.
Yeah, you guys are going to sit in these two seats?
No, I'll just sit over there. That's all. I'm first, but I'm done.
No, you have to sit up. Yeah, you have to sit up here. We're going to put that name right over there. We just wanted to make sure you were on it.
So I'll just...
And you guys have water, you're also...
Can I get, at least this water is mine or is it yours? No, it's somebody else's.
I think they left it here. Yeah, I'm going to pour all that out
Oh,
right
can
now.
you get maybe, this is yours?
You can do what they do in the street. Just take a match and reseal them. You'll never.
Yeah, that's right. That's a good one?
Yeah, that's a good one.
That's all you can do. Yeah, well, they do it on. They do it. Those guys that go into the tunnel too. Just why is it so thin that plastic?
Almost didn't get here because we were coming down from Connecticut, and all trains stopped when we got to Stanford. So there was some power line down.
Hey, Rick, can you sit down so we get started? Jesus Christ, I'm going to be on top of you guys.
I had to grab a Uber
Oh, really?
to get here.
So it's literally just stopped.
Nothing. We're suspended. That's what work was suspended.
Oh, there she is. This is like corralling cats.
I know. Tell me about it.
Or my children.
Either one. Okay, folks, we're going to get started now with the second panel. If everybody could take a seat, please.
That's good. That's your water, yes. Okay. Oh, no, if you want it, I was just asking. And I don't think, he said none of this is his. So I don't know.
I don't know whose phone this is. Okay.
Yeah, somebody will claim it. That's fine. All right.
Okay, we're going to get started now. The topic of this section is applicable affordable treatments, loosely based. All right, and if you guys thought you heard enough infectious diseases earlier, you're going to get a handful this time too. We've got two leading experts in infectious disease right here. So the first person who's speaking is Mr. Rick Flath. He's the founder and president of the Institute for Life Sciences Collaboration, which is responsible for this whole arrangement here. He's one of our leaders. Rick wears many hats. One of them isn't as a conductor of a train, apparently, because he came in on Metro North, and the Metro North train quit on him. And so they sat in Stamford, I believe, for an hour, waiting to get an Uber to take him in here. So he's had quite the trek. Just to let you know, Rick, I take Metro North all the time. So we're really proud to have him. Give him a round of applause to welcome Rick.
Great, thank you very much. Great. Thank you for the introduction. And yes, we can say that Uber works. And thank for some of the folks that was able to get that. Otherwise, we'd still be in Stanford, I think. Let me tell you about the Institute for Life Sciences Collaboration. We have three offices. Aaron is, wherever he is our secretary treasurer, and the fellow out in LA is our chairman. And what we do is, we basically are a group that looks at collaboration as the way to get things done. And we've been around for about 4 or 5 years. We have worked through many of these programs. The last one we did was on climate change and the impact on health, on children's health. We work a lot with the UNA, UUSA, and all the other NGOs. But I wanted to talk today about not so much what ILSC does, but really how we collaborate with the other groups. And one of the things that's always dear to everybody is how do we find money? So one of the other jobs I have is I'm also the chairman and founder of a private equity company where we invest in early stage drug discovery projects. And my first role in looking at raising money was almost 25 years ago. Actually raised money for a nonprofit in a little town called New London, Connecticut, where we raised $15 million for an old performing arts center. The town was going to knock it down, and we said that's crazy. The culture of the United States is built on performing arts. So we saved it, and it's going great. It's a very success. So one of the things I want to do is just break this up, and I only take 10 minutes, into two parts. The first part is I'm just talking about where's the money. This is from the was just published last year, 2016. There was $390 billion was given out by the good folks in the USA. $390 billion. Obviously, you can reread the chart. Much was went to religion and education. But what is key here is this is an enormous amounts of money. And in 2015, it was around $380 billion, or 375. So the first place, looking about where is funding for the types of things we want to do, we can't forget that as a country, this is incredible, that we as a country willingly produce about almost $400 billion. Now remember, on a donation, for many of that, especially in the foundations and so on, they have to give away to keep their nonprofit status 5% of their assets. So there's enormous amounts of assets that are out there, not just counting the amount of monies that is given every year. So keep this in mind that the key part, and we'll talk about how do you access this money, but there is, again, this is the latest information. The second thing is on where the money is on a public sense. This is just simply the National Institute of Health, NIH. NIH is run by the government. It traditionally gives about $32 to $35 billion a year out. It is what we call the government's venture capital arm because it funds well over 50,000 grants, well over 300,000 researchers, and 2,500 organizations receive money. Is there anybody here in here who's received a NIH grant? Okay. Trying to? Okay, great. Well, this is a source of money. It is hard to get to, but it is a source of money. I'll let you talk about that later. But it is enormous amounts of money for healthcare, and it's again what we would consider the government's venture capital money. These are the schools that get most of the money, and I find this pretty interesting. I just went through the 2,500 schools that, or universities, whatever organizations that pick up the money. These, I don't know, 20 or so, routinely get over $100 million a year. So why is that? Is because, I mean, this is not a definitive list. I just quickly went through 2,500 names and pick out what I basically the ones that struck me there. But what's interesting to me is that for NIH dollars, the NIH people look for organizations that have a real track record of success. Really hard if you are a independent person to get a NIH grant if you're not somehow or another connected to some of the top organizations here. But it's interesting to me that these organizations are very familiar with everybody. So what they're going to do is, I lost the key here. So if I'm Hopkins, okay, I get $347 billion, but then go back to this slide. So if I'm Hopkins, what am I going to do? I'm going to figure out how do I now pick up part of that $55 billion that's out there. And so when you look at where the money is, you have to look at how it's leveraged. And the former mayor of City of New York gives a lot of money to Hopkins. I think his last one was about $300 million. So part of this is these monies in here are leveraged in a lot of different ways. So the other question that always comes up from the private side is why, if I'm an individual, why do I want to give you money? Right? Not from the public side, but from the private side. Why do I want to give Barry any money if I'm a private equity person? Well, one of the things you have to consider is that from an investment point of view, the S&P Biotech Fund has decreased by 50% since 2015, and it's gone back up a little bit because the market's gone back up, but it's currently down 25%. So what does that mean for someone who's looking for money? If Barry came up to me and said, I have $100 for you, Rick, to invest, where would I put it? So I do a very simple allocation. I say, well, Barry, you got to put some money into the internet. You got to put your money in here and whatever. And at the very end, because of the difficulties that the biotech has, it's such a long-term investment, I'm going to say, give me the last $10 and I'm going to put a shot here. We'll take it a go. So one of the things that all of you who are thinking about, and Norman's question and Dick and so on, is how will these things solved? We have to figure out a way to turn the cycle around so it's not Barry's last $10, but it's his first $10 that goes in. But here's some of the reasons why the biotech stocks have reduced by 50%, but now down by 25%. First of all, there's tighter drug pricing controls. So why do you want to invest in a company that has cost controls? Unclear, well, Lord knows, unclear direction in the healthcare world. in healthcare law, unclear pricing policies, how that's going to affect pharmaceuticals, tax policy. There's a lot of money in pharmaceutical companies, but it's overseas. How do you get it back? And then the last thing is, pharmaceuticals have what we call terrible brand. Not a lot of people would say one of my favorite organizations is the pharmaceutical companies when I think someone's mentioned it costs $1 million to have a drug. So, so just generally speaking, it's hard to invest. And finally, all this leads to better opportunities for investors. So if I'm sitting on a lot of money and I'm a private investor, I'm going to think of all these things why I should not give you money. In fact, Barry's going to say, in fact, forget the 10, I'll give you $4.50. And the last thing I want to point out is the size of the market. This is very important. I'm trying to give you a realistic point of view here. So where does pharma rank in the top Fortune 500? This is the latest information. Johnson & Johnson is the largest pharmaceutical company, but it ranks 35th in size, revenues of $72 billion, profits of 16.5. Who do you think the largest company in the United States is? Well, no, I think it's Apple, right? So, Apple. So here's a company that didn't hardly existed 20 years ago, and it's now the largest company in the United States. Pfizer, we always hear about the great work of Pfizer, right? It ranks 54th. You start getting above 50, it's like, who cares? Merck, 69th. And just to give you an idea on the size of this thing, if someone said, do I want to invest in Johnson & Johnson or Merck, or do I want to invest in the companies that sell the drugs, where are you going to put your money? CVS, 7th in size, has $177 billion. Express Scripts, most of us get our drugs from Express Scripts, in mail. It ranks 22nd in size, the Fortune 500, has revenues over $100 billion. So these are the types of things that when you say, where is the money? There is money surely in NIH dollars, but very difficult. Remember, 50,000 people or grants are out there. That doesn't count the other 50,000 that don't go anywhere. You're competing with schools that routinely get $100 million a year, that list. They have a big influence on how they get money. You have the nonprofits, and then you have the fact is that pharma doesn't strike you as a place that I'm an investor where I would normally put my dollars. And so one of the things you want to look at is where did venture capital put their money in the last quarter, the first quarter, 2017. Top thing, $58 billion in one quarter went to the internet world. 3.7 went to healthcare. And what's going to happen in the next 5 years, the hot area of investments is going to be in AI and machine learning. So all I'm saying is the competition for biotech money and for your ideas is going to be very, very high. And then to Dick's point of view, so now Dick's talking about some pandemic in the middle of somewhere, and he's saying, Rick, can you lend money to it or give money to it, and see how hard that conversation is going to be. So the last thing I want to do is, this is from Canaan Partners. Canaan Partners is a group in Westport. I know of them. I have nothing to do with them. But if you go to their website, they have something called their entrepreneurial pitch book. And it's 20 pages of, if you're looking to raise money, they give you a kind of a diagram of how to find it. It's a very easy read. I don't have anything to do with Canaan Partners, but I found that their work that they did to do this was good. And they tell you, intro to the ask and so on. It's a standard thing. So anyway, I want to, first of all, thank you for your time. Thank you for coming today, and all my co-sponsors. And I will turn it back to Dave.
Thanks very much, Rick. I was interested, just Rick pointed this out, I actually was at Pfizer for 20 years as a clinician and retired from there. And now that I see that their Pfizer's 54th, they will probably buy Allergan or something to make sure that they can become #1 again. Yeah, right. So nice. Yeah, they keep on trying. Are there any questions for Rick right now?
Good.
All right. Okay. We'll get to the question round later. So. Yeah, no, you won't be. All right. Now for the next speaker, who's a very dear friend of mine, Steve Sholin. He's an infectious disease doctor, and he's, gosh, where are you now, Steve? You're either in Thailand,
New York
Philippines?
City, Dave.
Yeah,
New York City.
Is that where we are? Okay. So, yeah, Steve has a practice in Arizona and in Connecticut, kind of a long-distance relationship, and he also has a long-distance relationship with his wife, who lives in Thailand. So Steve will be talking about rabies, a topic that we don't usually talk about, and Steve is so dedicated to rabies that his email address, honestly, is rabiesfreeworld@aol.com. So go ahead, Steve.
Well, thank you so much, Dave, and thank you to everyone for attending. I am going to try to keep it on time here with the timer. Thank you. So this sort of changes things just a little bit, but my objective today is to show you one of the worst neglected diseases, rabies. And I know we've got a diverse audience today. Maybe some people have actually seen human rabies. But this is Bernard. He's an 8-year-old kid. He was bitten by a dog 7 weeks before he came to our hospital. And he actually had a few symptoms of fever, some agitation a day before. The sad fact of the matter, the dog died, and the parents took the child to see a doctor, but the doctor didn't think to ask about rabies. So you can see the child is quite agitated. He's moving around. He has some heavy secretions. They call this hypersalivation, the thick saliva. There's some irregularity to his breathing as well. Now this is the telltale symptom of rabies, the hydrophobia. So you can see that remarkable symptom that is pretty much unique to rabies. And unfortunately, 30 minutes after I filmed this, the child died. That's one of our other diagnostic tests, is the FAN test. And all of this took place at a hospital in the Philippines. It's actually our infectious diseases referral center to the country. Wonderful place. I always feel like I'm at home when I visit there. But they do have real problems like elsewhere in the developing world. And certainly they are trying to do the best they can. Some of the points that Dr. Gray made earlier about just do it. You know, why does rabies continue on? Why do we let this happen? When I was making rounds in this hospital, I just thought, oh my God, you know, is this the dark ages? How can this go on day to day? And here at this hospital, they actually have a rabies ward. So they have, you know, 1 or 2 patients coming in per week with clinical symptoms of rabies. And that sort of makes me want to do more, and that's why we have the rabies-free world, to try and raise awareness. But I think really rabies is the worst of all the neglected tropical diseases. There's so much suffering going on, so much death, and can't we do more? This is their animal treatment bite center at the hospital I worked at. They see many, many hundreds of bite victims per day coming in. A lot of them are just children. And if it's not treated, what could happen? You could develop rabies if you're not given the prophylactic treatment. It's actually the most lethal of all infectious diseases. Even with Ebola, you have a 20% chance of survival, but with rabies, you have a 0% chance. And this is a nerve cell showing the viral inclusions. And certainly rabies has been with us for a long time. It's been with us for hundreds, if not thousands of years. We think this virus is the most important of viral zoonosis in the world. And this is because the sad fact is that children are dying from this. About 40% of the cases are under 15. And a lot of this is due to dogs. Dogs in other parts of the world, you see here in New York City, dogs are in the pocketbook and that kind of thing. But dogs there are sometimes left to themselves. They're roaming the streets, feeding on garbage, that kind of thing. And an important factor when you think of worldwide rabies. Certainly in the US we have rabies, mostly it's wildlife. And that's considering the bat as well, which had some brief mention. But what we realized in the 1930s and 40s that if we vaccinated our dogs, we would take care of our human rabies problem. And certainly went from about 50 to 100 human cases per year down to a baseline 2-3 cases. Some of these are imported, some of these are bat-related cases. But again, I think thinking about the dog bites, the animal bites, these exposures, trying to protect people, knowing clinically what we should know as physicians and healthcare providers about screening for rabies, what do we look for so that we don't miss any cases, and just being aware of some of the exposure risks, because rabies is a neurotropic virus, so it's going to look for nerves. Obviously, some parts of the body have more innervation than others, and therefore could be at higher risk. One of the interesting things about rabies is it has quite a variable incubation time, anywhere from 3 weeks to 3 months. But believe it or not, some cases can actually occur more than 1 year later. And we know this because some of our immigrants who come from different countries in the world, they may be living in the United States for a while, and then suddenly they develop clinical symptoms, they have rabies. We can take that virus, we have the tools now to see where it came from, such as Philippine dog strain, Thailand dog strain, Africa bat strain, you name it, and we can figure this stuff out. There are the typical symptoms of rabies, which you saw in the video: hydrophobia, foaming at the mouth, agitation. But basically, any neurological symptom can be present. There is something called paralytic rabies as well that may be somewhat confusing. And the sad thing is, in the United States, 2/3 of infectious encephalitis cases, we never figure out what they're from. And you hate to think of maybe a transplant case. I know we've got some important hospitals and health centers here, but there have been, on more than one occasion, a person dies, the diagnosis is never made, the organs are transplanted, and guess what? The gift of life turns out to be the unfortunate gift of rabies, and inevitably these patients die. So I know our theme is affordable treatments, thinking about repurposing of drugs, but unfortunately since the 1880s there haven't been many innovations. There's no actual treatment when you have the clinical disease of rabies. What do? You basically sedate the patients and you watch them die. So I know there's a lot of talent in the room just to light the fire under you, thinking about what else could be done. Now part of this stems to the virus itself. It's very tricky what it does when it attacks the human body or other mammalian nervous systems. It basically hides within the nervous system that's protected from the immune system, so you don't have the immune response, and that allows an overwhelming infection to take place in the brain before it's too late, before the body can do anything and take care of it. Now we do have, for anybody who's in the medical field, we have standard precautionary prophylactics that you want to implement before you develop symptoms. But again, if you have symptoms, there's nothing that can be done. So I think there should be a multifaceted approach, primarily education, and you think of the individual getting the information out. Also at sort of group levels, could more be done? And the WHO has actually granted a World Rabies Day, which is September 28th. If you want to have a dog walk in your community or some sort of thing, we would appreciate that. Certainly on the medical side, we try to protect patients. We have immune globulin, we have vaccine, that sort of thing. And could we do more as a global community? And thanks to Bill Gates and all the donated monies that have done so much good for the top 3, particularly HIV, malaria, and TB, but what about rabies? And if you think about it, we could actually stop rabies. In fact, one of the WHO goals is to have no more human rabies death by 2030. Unfortunately, knowing what I know about this, I'm not sure that we're really going to get there unless we move ourselves and try to get to the point. So I think about kids, I think, gee whiz, what if that was my kid? What if that was somebody I knew? Couldn't we do more? Because nobody should really have to die from rabies. I think that education is huge, trying to get that momentum and trying to finish off rabies with vaccination. We can do it. So thank you very much. Hopefully that was 10 minutes or so.
I'm being informed we're not taking questions right now. We'll take questions later on. We'll get through the talks. That was a great speech there. 9 minutes, 44 seconds. I don't know how he did 40 slides in that short a time. I don't get it, with 3 videos. But you can definitely see the passion in Steve. And have you made a difference in Philippines, do you think, with you and your colleagues? Yeah. Yeah. Well, we need more people like Steve Sholin, that's for sure. Thanks, Steve. The next speaker is Dr. Barry Craisworth. I'm trying to figure out how to introduce you, Barry. Barry, I've known Barry for many years, more than we'd like to remember. But I think when you think about public health, especially in infectious diseases, his name is right at the top of the list. We're very valued to have him here. He's a true scientist in infectious diseases. He has a capital S for scientist. So welcome, Barry. Thanks.
Thanks, Dave. So just to answer the question, if you submit 100 grants, basically 10 get funded. So we're funding at a percentage of 10%. So 90 go down the waste. Okay, I'm going to talk to you about something you do know about, which is antibiotic resistance, a problem that basically we're in trouble with, and one that I'm not sure antibiotic era, which I'm going to tell you about how it started, but I also think it may be at our end. So let's look at the beginning. It actually starts back not that long ago. We're talking about 1940, a policeman in Oxford who comes into the hospital with a facial infection. There's no antibiotics then. They basically drain the infection. They use something called sulfapyridine. Over time, it doesn't work. The Staph aureus, which is on your skin, starts spreading, gets into your lungs, gets into your bone, and now now we're in trouble. Now we end up going back. Come on. So now penicillin, we're about almost 4 months later, patient's still in the hospital. You can imagine what it costs to maintain a patient that long. They try this new antibiotic just made called penicillin, 1941, 5 days of treatment, remarkable response, no more drug, patient dies, but it's the first indication that the antibiotic has started. We have now penicillin, and we can treat staph infections and other things, but the problem is as follows. We started using penicillin in 1944. Hammersmith is a large hospital in England. It's sort of like Bellevue in New York, and you can just see how fast, after no drugs, 1941 we start using penicillin. 1946, 13% of the Staph aureus don't respond. By 47, 38 don't respond. By 1948, 7 years later, 60% of the bacteria of Staph aureus now are penicillin resistant. This is the theme that repeats itself over and over and over again. And here's just a perfect example. This is a pipeline showing how remarkable the drug companies have done since then. We make these very, very famous drugs that you even know about, penicillin, tetracycline, drugs that are used in the hospital, Levaquin, which you know about. And so this is a remarkable pipeline. The problem is that the bottom pipeline shows how quickly the bacteria develop or adapt and become resistant. So ultimately what you have is you have the pharmaceutical company trying to stay ahead of the bacteria developing resistance. And this has been a constant war that's been going on, and unfortunately, as we'll talk about, the war is coming to an end mainly because the drug companies have basically stopped making antibiotics. So because of that, we're starting to get things in the press, various other discussions, which is talking about how antibiotic resistance is going to be a major problem. 2 million people each year acquire a resistant organism. About 100,000 of these people in the U.S. get that organism in the hospital. It's called a nosocomial infection. You go into the hospital for a gallbladder procedure, and you end up getting an infection in the hospital, and when you do, That bacteria in the hospital is very drug resistant and will almost be to the point where you can't treat it. That's the problem. 23,000 people die from resistant infections, and that number is going on. This is in the U.S. Now, someone mentioned this before. I think this is a WHO report, but ultimately what we're looking at is by the time we get to 2015, more people will be affected by antibiotic resistance than cancer, costing about $100 trillion. I think whoever said before, once you start putting numbers to these problems, people sit up and think, although it's hard to even think about what $100 trillion is. Okay, so as a result of this, both the Obama White House then put out a white paper on antibiotic resistance, and WHO has done the same. And the bugs that they now talk about, this is priority one, and these are bugs that have a fancy name, Enterobacteriaceae. What they are E. coli, Klebsiella, bugs that are in your gut and in my gut, but now they've become resistant to the point where we almost can't treat them. Why? So the problem is complicated, as our new president said. Who knew it was this complicated? But it is. The drug discovery pipeline is limited, and we'll talk about this because Big Pharma has gotten out of the antibiotic business. The population who we're treating is highly vulnerable. So the HIV epidemic is not over, but it's under control. We have a lot of immunosuppressed HIV patients, but we now make patients immunosuppressed because of our remarkable medical procedures. We can do transplants. We can do bone marrow transplants. We can keep cystic fibrosis patients alive, but they're immunosuppressed. They're purposely suppressed, which means they're vulnerable to infections. So now you have a lack of drugs, a highly vulnerable population, and bacteria who have developed remarkable resistance. Here's the spectrum. So penicillin, 1941 resistance, we develop a new drug, another penicillin, which works against that. Become resistant there, we develop the next one. Becomes resistant, we do the same thing. This is a continuum. The drug companies did a good job getting on top of it and always having that next fish to gobble up the one before. The problem is that when we got down here, carbapenems, which was back in the late 80s, were a remarkable drug. We're at the bottom of the pipeline here. There's nothing new coming. We're really groping. We have things called beta-lactamase inhibitors, which aren't that effective. This pipeline is drying up, and that's the problem. So what I always think about, this is literally the game of spy versus spy. Drug companies make a beta-lactam, the bacteria develop the enzyme that chews it up. And this is the continuum we see every day. There's more than 1,500 of these enzymes that the bacteria have acquired. They're very smart. I'm going to show you one example. These are the carbapenemases. They come in lots of flavors. So not only do we have one problem in the US, we have problems in Asia, the Middle East, and Europe. This is also in the Middle East. The biggest problem about this type of resistance, which means you can't use the drug, is that the resistance themselves are movable. They're on mobile elements. They spread from one bacteria to the next. So not only are you controlling the bacteria, you have to control these elements that actually move resistance, which we don't know how to do. Okay, this is a terrible slide. Whoops, the only reason I'm showing it is to tell you that anything in yellow, when you have one of these genes in a bacteria, it eliminates this entire class of drugs, which are all that I showed you before. So if I have a bacteria that has a KPC gene on it, I can't use any of these drugs. And those are the drugs I want to give the patient. So that's a problem. Okay, global epidemic. This is what it looks like. This started in about 2010. It is now spread literally around the world. This is not a U.S. problem. This is a problem in Europe. This is a problem in South America. The epidemic started right here, New York, New Jersey. Patients then traveled, visited people in Israel. We had a lot of exchange of patients that went from New York City to Israel. The epidemic started in Israel. It went to Greece. It went to Italy. It went to South America. We have another epidemic with these light greens, which is starting basically in the Middle East. We also have another strain that's starting in India, Pakistan, that's spreading. This is 3 epidemics that are crisscrossing around the world, and we can't treat these bacteria, and they're infecting patients who are very, very sick and very, very expensive to treat. Transplant patients is the new HIV in the hospitals. They are expensive patients, and they're highly vulnerable. Okay. So this is the ones we talk about in the U.S. But behind the scenes, we have something called tuberculosis. We don't think about tuberculosis. It's the other bacteria I work with. It's caused by a bacteria called Mycobacterium tuberculosis, related to Mycobacterium leprae, which causes leprosy. Very famous, very historical. Biggest problem is the bacteria spreads when you cough. It's an airborne bacteria. So you can be in the wrong place at the wrong time and get infected. That's the way TB spreads. That's why TB is not going away. You will not believe these numbers, but these are real. These are 2017 sort of numbers. Take them all with a grain of salt. They're underestimates. 1/3 of the world today is sitting with tuberculosis in their lungs. 1/3. 8 to 9 million new cases each year. 1 to 6 million deaths. This is the one you'll remember. Every 20 seconds, someone dies of TB. Today. Every 20 seconds. So since this conference has been on, there's been many people who have died of TB. The problem is, it's not here. It's in developing countries. But we need to deal with TB here, mainly because TB is a disease that's associated with HIV. And if it wasn't because of HIV, we still wouldn't have money to treat TB. And if it wasn't for Gates Foundation, we'd have no money to treat HIV or TB. Well, that's not true. HIV will still be funded. But TB really is now a disease almost solely funded by the Gates Foundation, both for vaccines, drug discovery, getting drugs to people. So this is a problem. On top of this problem, though, TB has become highly drug resistant. So again, another problem with resistance. But look at this. This is from 2012. I haven't seen a more recent report, but this gets to the whole conversation we had this morning, or this afternoon, earlier this afternoon. Here are the leading causes of death globally. Someone said about diarrhea, this is children diseases, but over the age of 5, the leading #1 disease-causing infectious is TB. It's right here. It's a bacteria. None of these are bacteria. This is a virus. These are viruses. The #1 killing bacteria in the world is tuberculosis. Look on this side. How many drugs have we made? Well, we've done a remarkable job with HIV, 70. Look what we do with cancer. Look what we do with TB. 5 to 8 drugs. 5 to 8. That's it. That's the whole pipeline. #1 infectious disease in the world, 5 to 8 drugs. This is what it looks like. This is from 1968. This was the last drug made until 2013. This was made by a company then called Letterly, which then became Wyeth, which then became Pfizer, as one company chewed up the next. #1 killer of the world for people over the age of 5, and we didn't make a new drug for 40 years until 2013. And the reason is there's no political will to put money into a disease that really doesn't affect us. There's about 9,000 cases of tuberculosis in the U.S. Most of them are foreign-born. But we have to do something with it because 1/3 of us are walking around with this, and a good 5 to 7% of those people will come down with disease, and when you come down with disease, you spread it. That's why we'll never get to cure TB. We just have to be able to control it. So why? Why do we have this problem? It's a slow-growing, highly infectious airborne bacteria. It requires very expensive facilities to work with, which I'll show you, which is what this slide's here. To work with TB, you need to be in this type, this is my lab, you need to wear this type of equipment because it's an airborne disease. So you have to be gowned up. This is called a BL3 facility. Very expensive to run, very, very high-risk work. My people who work there are very dedicated, and I have a lot of respect for them. Drugs must be made in combination with current drugs. You treat tuberculosis patients with 4 drugs. So if I develop the next perfect drug, but it doesn't work with those other drugs, I can't make it. On top of that, I can make the best TB drug in the world, but if it doesn't work with people who have HIV, meaning if it's not compatible with all the different HIV drugs, it's not an effective drug. So that's why the drug company, you got to be crazy to try to make a new TB drug. Not to mention it's going to take months to years to grow because the bacteria grows slowly, and clinical trials are going to take tens of years. The real reason, though, no matter what anyone tells you, is because it's in developing countries. And that's the crux of the problem. We need something to incentivize. We need ways to get money. And like I said, we're depending on foundations like Gates Foundation, which is wonderful. They've done a great job. but they shouldn't be the one that has all this burden. It really should be a global job. So I want to leave you with this thought, and I actually believe this, and the longer I work in this field, I believe it more. So the antibiotic era, which began with the first use of penicillin in 1940, is rapidly coming to an abrupt end as a consequence of a sparse antibiotic pipeline and an evolved microbial population that harbor a diverse and mobile array of antibiotic resistance genes. Unless we collectively approach the problem with the urgency and commitment that drove the Manhattan Project, that's when we made the atomic bomb. I lost my last slide, oh no. The point is, we're not going to basically solve this problem. We need that type of commitment, and that means commitment from everyone. And I'll stop then.
Thanks, Barry. You know, when I was a fellow, an infectious disease nephrology fellow back in Minnesota, I had a patient, this is 1981, had a patient that had Pseudomonas infection that was resistant to ciprofloxacin and imipenem. Amazing thing is those drugs weren't on the market yet, and it was already resistant, which highlights one of my reasons around resistance is if we can take the antibiotics out of animal feed, that's one of the biggest burdens of our sources of resistance in the globe. We can't control that, unfortunately, yet. So nice job, Barry. Now, our next speaker is, and I'm sorry about your name, I'm going to bastardize it, I'm sure. It's Dr. Corey Troselje? Pretty close? All right. Corey. So Corey, she's a molecular oncologist with significant contributions in the emerging field of epigenomics. And importantly, she's also a Fulbright scholar. So welcome, Corey. Looking forward to your talk.
Thank you. It will not be easy to speak after these wonderful speakers, especially not because English is not my maternal language. So I'm coming from the country which is pronounced to be one of the best world's destinations for this summer. Welcome to Croatia. I will speak about personalized approach to cancer therapy because this is disease that I deal with for 25 years now. So when people ask me what the cancer is, just to put it in a normal word that everyone can understand, it is a story about the cellular division. Normal cells want to make daughter cells, and they do that through a very, very strictly controlled process, which is called cellular cycle. As you can see, at the end of the cycle, we have two daughter cells. Cancer cells do not want to follow this rule. Cancer cell is all about immortal division, and uncontrolled cellular division, which makes them immortal. That is abnormal. If we start the story, and I'm coming from Europe, we love speaking old stories. So the first mentioning written on cancer was 3,000 years BC. The paper is known as Edwin Smith Papyrus and describes 8 cases of ulcerous breast cancer with the final decision there is no treatment for this disease. I show this because I'm in New York. Actually, the paper is in New York. It is kept in the New York Academy of Medicine. Cancer, according to data released by World Health Organization in 2015, says that 41 million people diagnosed, 80.7 million died, 32 million had to live with cancer. That tells that this is a chronic disease. And finally, in 2030, it is estimated that more than 12 million people will die from this disease. When we take a world map, this is not a good situation. United States, Australia, Western Europe, highest incidence of cancer in the world. The same count for females. If you take a look on age standardized rate of cancer, then we will see this very, very high incidence of cancers in females and very low incidence here. The first one is Denmark. The green one is India. So the question is, why Denmark, which is a very developed country with excellent healthcare, has so many cancer cases. It is not only about incidence, it is also about mortality. That is the country with the highest mortality and adds to this red European map. We don't know. What we know is that we don't know. But we do a lot of research. The Cancer Genome Atlas is a new, relatively, I would say, new project which represents a collaboration between the National Cancer Institute and National Human Genome Research Institutes, analyzing so far 33 types of cancers, and deposited 2.5 petabytes of data, and petabyte is, I've learned that recently, 10 to 15. So what do we do with this data? How did we come to this data? This is actually extension of a human genome project, a huge project in human history, which had many tasks to identify our genes, to determine the sequences of DNA, to store this information in databases, improve tools for analysis, transfer technologies to the private sector, and address ethical issues. And there are many of them, and they still are present and live with us. I will show that a bit later. So it was Manela Esteller, one of the leading names in epigenetics, who says, we have this huge phone book. It is a phone book. But it is the time to organize it and make some calls so we can really be sure that the names and addresses correspond to this number. In other words, what does this data mean for us? What the consequence is if we have so-called mutation? I like this slide. Took me several hours to make it. If we say that it starts on DNA, that is correct. DNA must to be replicated in order to double the material, which will be divided in daughter cells. But DNA also must give a template for the RNA molecules, which are a sort of mediators between DNA and proteins. All of these molecules make us interact on, and all these molecules actually communicate on many levels. When we speak about our genome, genome relates to DNA. Genome and cancer, we know that 70 to 80% of cancer represent sporadic cases. Only 5 to 10% represent so-called hereditary form. What's the difference? What happened? Okay, in sporadic forms, the person have two normal genes, we say alleles. During development, as we become older, one gene may be damaged. In that case, the cancer will not develop. It is needed for both copies to be damaged, and then the tumor develops. In inherited cancer, the child is born with one damaged gene. So the window for the other mutation to happen is relatively short, and this is the reason why inherited cancer really occurs in an early childhood or younger age. Some inherited cancer syndromes are listed here, and I thought that for this audience maybe the most important would be this BRCA1, BRCA2, which is highly publicized and people read about it a lot. But BRCA genes are also very important because they brought us to a very, very sensitive field about gene patenting, which was related to the company called Myriad. And a big lawsuit which was brought against Myriad by Association of Molecular Pathology. So what was the problem here and how it was solved? Importance of legal action put in question the validity of gene patents in the US because the owner of the patent owing the DNA sequence claimed that he, the owner, will develop diagnostic methods as well as the method to identify drugs using this owned DNA sequence, which means that others are not allowed to work with it. Luckily so, naturally occurring DNA sequences cannot be patented. So that verdict was brought 4 years ago. What is personalized medicine for cancer patient? I put here 2 definitions. By European Union, it means providing the right treatment, right patient, at the right dose, at the right time. NIH gives quite broader definition, which I like better, claiming that it is a form of medicine that uses information about person's genes and proteins and environment, not only to treat, but also to prevent, to diagnose and treat disease. Now, how different are we at the level of DNA? Are we different? We are similar, yet we are very much different. So there are databases related to Human Genome Project, which are updated every day. One of them is so-called a single nucleotide polymorphism database, which claimed more than 112 million polymorphism in humans in 2014, close to 150 million in 2015, and I checked the other day, it lists more than 336 polymorphism in human beings. And what does it mean? It just means that person A on a position on a chromosome or in a gene may have A or C or G or T, and this is how we are different. It does not mean that we are sick because of that, but it clearly means that these differences are of utmost importance for making us susceptible for getting disease, as well as for influencing our response to therapy. So what is the idea of personalized medicine? First of all, we must know how our normal cell react, and what is there on its surface. When we know that, we may check malignant cells which really can have similar things on its surface, but they are acting in a sort of devilish way. Ideally, when we define it, we want to neutralize it, or we want to completely close the window where these things act. For example, half of the melanomas, tumors that are very common in this country as well as in Australia, have specific mutation in the gene whose name is BRAF. This mutation forces cells to grow and divide quickly. Now, when we know that there is a specific mutation, we are going to make a specific drug that will bind to that place in order to close it. As shown here, this is our first published result. Their tumor volume in animals that were receiving the highest dose of the drugs did not progress. It looks from a perspective from people who are doing this so-called protein modeling. But cancer cell can also have on its surface things that actually normal cells do not have. That's the other aspect that is also very well recognized, and what we also want to block it. In other words, these proteins which are typical for cancer cells can be and should be blocked, and at several levels in order to prevent what just heard about bacteria, to prevent them to develop a multi-resistant phenotype. Different cancers have different frequencies of mutated genes. So if we consider this slide here, there are different genes that can be changed in cancer. And for this one, KRAS, you will see this blue column represents pancreatic ductal carcinoma. APC is the most commonly mutated in colon cancer, etc., etc. What came only a few days ago is that we got the first approval for any solid tumor with a specific genetic feature, which does not relate to the origin of the tumor organ-wise. So there is this specific genetic mutation. Give that drug, doesn't matter where the tumor is coming from. So it is not breast-specific or colon-specific or pancreas-specific. So this is a study that actually was very important for FDA to make this decision. These are how many mutations are there per malignant tumor? Enormously much. And if the tumor cell cannot make repairs during replication, these numbers become extremely high. Here is the rank 9, here is rank 3 that should be recognized. How much should we pay for performing the test? Affordability question. When we started sequencing molecule of DNA, the cost of megabase, which means 1 million nucleotides to be detected, was around $10,000. It dropped significantly in 15 years. How much does it take for sequence the genome? It started with $100 million and now is here somewhere around, I would say, 2,000. It's doable. This is what Human Genome Project, with all the methods that was developed, actually did for us. Now, what do we know about, how do we understand this? So this is example of pancreas cancer. Take a look. Normal cell can get one mutation, the second and the third, and it still, at this state, will not proliferate like crazy. It will be stopped by defensive mechanism of the host. But when it comes the third or fourth, it will start to proliferate. Will that cell produce identical clones? No. All these clones will actually become specific by themselves. And when we have a drug to react here, and if we recognize here, we are killing only this, we are not killing that. And this is actually why we fail to treat tumors with so-called monotherapy. We need more drugs involved. The most successful story is Herceptin story. It is drug taken to treat breast cancer, identified in breast cancer in 1979 and presented in 1998. FDA granted and approved Herceptin for treatment of HER2-positive metastatic breast cancer in September of that year. It's not only that. What is extremely important for personalized therapy is that in parallel with the test, the device or method to detect mutation must also be approved. The essence of personalized medicine is co-development of gene-based therapies in combination with a test to detect the drug targets. I will just show that it means the right therapy for the right patient. There is, of course, always the question of affordability. So it's estimated when Roche, a huge pharma, put Perjeta on the market, it was estimated that it will increase the survival of women with advanced breast cancer by 15 months. However, the treatment would cost in Great Britain around 43,000 pounds. So it was the question who's gonna pay for it, and I'm not the one that will answer this question. These are questions that can be asked. I will skip this and show you that you, by yourself, can go to Clinical Trials Gov website and check the clinical trials of specific drugs if you want to really find something more about this. These are shown for Iressa and Perjeta, 2 drugs, and look at how this number increased in only few months. This is before they were put generally on the market. Now the number of studies is decreased. At the end, I want to say that real world taught us that we do not follow the rules, and it came out that in this country 76% receive smart therapies without being tested. In other words, these methods are not used. It's far better in Asia, but basically the rule should be that everyone who is a candidate to receive a smart drug should be tested for that. This clearly shows that we are still not completely aware of everything that we can do. I will skip this. Post-market surveillance is for that reason very critical to the success, because we can fail on indeed many levels, including to design deficiencies, manufacturing deficiencies, or operator error. This is what I see every day. And I just wanted to show you these top 10 best-selling biotech drugs in 2014 in the world. And what we can see is that cancer drug Avastin is on the 6th, Herceptin is on the 7th, and Neulasta, which treats chemotherapy-related infections as a side effects here, is already on the 8th place. This is the number of smart drugs that are approved per year since 1994. And present and future challenges should definitely think about limited understanding of the intrinsic biology of cancer, common conditions involving multiple genes and biomarkers. It's not monogenic disease. Our outdated disease classification system, our lack of infrastructure, our problems in clinical implementation of new diagnostics, as we just saw here, investment uncertainties, and access to personalized therapies. We have absolutely many things to think about, as we should, as I put as the last slide, think about epigenetics, which is how genes are regulated, because this is about more than genetics. This is how it all works together. So when we put all this together and we do smart things, we may do good legacy for the future. Thank you.
Thanks very much, Corey. That was a great talk. Appreciate it. We have a little bit of time available for some questions, if anybody has any questions. Yeah. Rick, you want to come up? Well, I have a question, Corey. I came from watching TV last night, and I watched an ad, I gotta remember what it's called, 23andme.com, which for only $199, I can get my genome discovered, kind of like where you can, you know, adopt a manatee down in Florida or adopt a star. I guess the question is, if we look at somebody like Steve here, fine young man, healthy as a horse, is it necessary for him to get his genome sequenced?
It depends on Steve. We must be aware of what we can do, and we must also understand why we do something and what are we going to do with the test result. So there are many biotech companies that will do these things for profit, and Steve will get results as a big sheets with how his genes are put together through all these letters. The question is whether he wants to do that, and if he does, what is he going to do with that? I haven't had my genome sequenced, nor I will.
You have not?
No.
Okay. That answers the question right there. Okay, thanks. We have a question? Go ahead.
Good afternoon. So my question is for Dr. Steve Scholin. So many years ago, we did a campaign to eradicate polio. If rabies is more devastating, why haven't we been able to do the same?
That's a great question, and how fantastic it is to have eradicated polio from the Western Hemisphere. You know, still trying to close that virus down. It's proving very, very difficult because it's so wily as a virus. You know, I think the good news for rabies is that at least we've taken care of the dog problem. And so in some ways it's a relatively simple solution. Vaccinate the dogs and you can control a disease to a large part, except for some of the wildlife and the imported cases. But other places around the world have done this. If you look at Latin America, Mexico, they realized, you know what, if we start immunizing our dogs, dog control, we can take care of the human problem as well as the animal welfare. I think part of the reason I wanted to present to you, and thanks for listening, was that, you know, rabies is on that list of neglected diseases, and there's so much that needs to be done, And where are our priorities? You know, Dr. Gray was advocating, you know, just get out there and do it. I mean, what are we spending all this money on? You know, when you look at GDP and all those trillions of dollars that are in play, what about health priorities? Wouldn't it be good to invest in that? So, you know, raising awareness, I think, goes a long way.
Great, thanks, Steve. We have a question in the back here. Why don't you wait for the microphone? She's coming around with it.
Thank you. I have a question for Dr. Corey. It's about awareness of breast cancer and epigenetics and genetics. In India, we never had breast cancer. The only population that had breast cancer was the Persian migrants who had migrated thousands of years ago. And we knew they had the BRCA gene and they got it. But the Indian women never got it, and yet now we have, in the last 25 years, it's a rising problem. And lots of Indian women, not only are they getting it, they are dying from it, and we have to take care of it. And we never knew that they would get breast cancer earlier, so no one diagnosed them with it. And suddenly, all of a sudden, now that we know, they're getting the lump and they need to be. Now, in one household, we had the owner of the house and the maid who got breast cancer at the same time, the same year. So obviously, there is an epigenetic factor that's involved here. We don't know how we would go around getting the poor people involved, but the maid's boss got the best treatment. The maid's, her breasts were both cut off, and you know she was taken care of that way. Now the poorer people in India will not get diagnosed. We don't know how many are dying from breast cancer, but we have a lady who's deeply involved with the slums, and they're saying stomach cancer is the biggest problem growing among the slums of India. And they're asking everyone's help on how to solve that. They're saying, can you please bring awareness to the problem? We do not know why the slum children are getting, adults and children are getting stomach cancer. If there's any way to look into it, there's no funds, naturally. And then if you are diagnosed with it, there is no cure, because who will fund the cure for the poor people of the slums? So these are my questions.
May I? So whatever you say is painful, I think, for everybody in this room, because I'm not sure that there is one single family in which at least one member do not have a cancer, and I'm not an exception here. So speaking, what I showed here, this data for Denmark and India, indeed shows that India is still far lower, speaking about incidence, than other countries, and but the thought is that it relates to lifestyle and nutrition. Lifestyle and nutrition, because I think that people in India genuinely eat much healthier than we do as a part of our so-called Western diet. It is well known that obesity goes well hand in hand with concretely breast cancer. It is also known that cholesterol makes a nucleus, which may be a starting molecule in estrogen synthesis. And estrogen is, again, the hormone that stimulates this growth. Are we exposed to exogenous hormones? Absolutely, because they are hormones that are used for animals to grow fast in order to decrease expenses that are needed for market for somebody. So I think that that the kind of life is changing as a part of global life change in India too, and that probably is part of the reason for this increase. There was regarding the gastric cancer. There are studies. The gastric cancer is a huge epidemiological problem in Korea. And when I was there, I recognized that they eat enormously hot and spicy food, which I adore. But even for my half-Hungarian stomach was a bit too much. And then I thought to myself, is it possible? Now, this is a speculation. I have no scientific proofs for it. Is it possible that this hot food makes small, like we say, micro-traumas? And then when the bacteria is present as Helicobacter pylori, that it basically cuts the barrier between the host and bacteria. That is thought, I never saw the study that really performed something like that. So maybe the food, and you said it's in slums, that could be maybe a lower immunological response and immunosuppression for whichever reason, and nutrition may contribute to what you addressed as a problem.
We have several questions, so if we can keep the questions short and the answers short, we'll be able to get a few more in before 4:10. The fellow at the back here, Dr. Gray.
I'm not going to ask the question, but the lady who made the premise about cancer and the incidence of cancer in India is incorrect. Come see me afterwards, and I will show you that cancer has been in India for a very long time. It's a cultural thing.